Hello everyone, and welcome again to criminal investigation, homicide investigations. Today we are looking at biological evidence and the scientific analysis of biological evidence, as contrasted to what we'll be discussing next class, which is non-biological evidence, right, and its scientific analysis. So let's jump in. So if you recall, we looked at evidence in a previous class and we grouped things together by typology, right? And so one of the groupings of evidence that we looked at was real evidence, right? Real evidence is that tangible thing you can touch, you can hold in your hand. That's kind of be our focus for this course, or this class and next class. But there are some types of physical evidence that don't necessarily fall into the real evidence category, and we'll talk a little bit about those as well. That being said, when we think of physical evidence, right? So it's there, right? It exists. It's not testimony, right? It's not just words, it's not confessions, but it's an existing thing. We can break them down into really 6 categories, right? So we have biological evidence, which is going to be the focus of today. We have impression evidence. So think of things like footsteps or footprints, tire tracks, things along those lines. Trace evidence, so really the fine details that you miss. If you're not careful when you're looking. Trace evidence includes things like paint chips and very small pieces of evidence that can link somebody to a crime. We also look at digital evidence. So we're obviously in the digital age, and more people are using the internet as a part of their crimes. And so we have to take a look and see how we deal with digital evidence. Then we have question document evidence. So this is evidence that's from a kind of document and whether a signature is legitimate or not, and things along those lines. And then we have narcotic and illicit substances, right? So we're talking drugs for the most part. That being said, today's focus is going to be biological evidence. And then in the next class, we'll discuss impression evidence, trace evidence, digital evidence, question document evidence, and narcotic/illicit substance evidence. So if we look at biological evidence, we'll see that there's kind of 5 really big things that stand out. Right? So when we're looking at biological evidence, and this is true of really any crime, but in the context of a homicide, we're looking for blood, right? Usually that's going to be, if there's a homicide, that's kind of a given. We'll look at semen and the notion that if there's a sexual assault, what gets left behind, how do you detect it, that sort of thing. And again, if it's related to a homicide, semen does come in handy. We'll look at saliva, right? So basically the wetness of your mouth. We'll look at hair, and we'll note there are some problems with hair evidence. And then finally, we'll kind of wrap it up with just a very high-level discussion of DNA evidence, right? And DNA is kind of found in the predicate blood, semen, saliva, and hair. So let's jump in. All right. So when we think of biological evidence, right? Biological evidence, the types, it's exhaustive. It includes blood, semen, saliva, vaginal secretions, skin cells, hair, perspiration, urine, feces, vomit, you name it. Right? If it comes from basically the human body in some way or another, we kind of typology, we put it as biological evidence. Now, that being said, when we look at crime scenes, the most common things that we find are, especially in homicides, blood, semen, saliva, and hair. Now we're going to look at each of these and see that there's kind of problems with all of them. by themselves, but taken together, they can form enough to make probable cause or even beyond a reasonable doubt. That being said, what's omitted from this list is the most common piece of evidence at a crime scene, and that's skin cells, right? So you shed hundreds of thousands, if not millions, of skin cells a day, right? You're probably shedding them right now and you have no idea. They are tiny and microscopic. Yes, they can give us a DNA link, but the problem with skin cells is they are everywhere. So the investigators rarely look for skin cells, because, I mean, think about a house, right? Let's say a murder occurs in a house, and there's 4 people that live in the house. And yeah, they keep it relatively clean. You're still, maybe you find a body in the living room. In that living room, you're going to find millions and millions of skin cells, right? And yes, maybe some will belong to the perpetrator, but most of them are going to belong to the people that live there. And it's kind of a question of time and resources. Do you really try to go after these skin cells and sort through millions of them? Or do we look at more obvious evidence, evidence that is a little bit easier to handle and then financially easier to deal with? All right, so that's where blood, semen, saliva, and hair come into play. So when we look at blood evidence, right? Blood evidence, we most find blood at homicide scenes and batteries, right? Now, this is a problem that I always have with people, or at least I have with the notion of the term battery. So when people say I've been assaulted, right? Most states, it depends on the state, but most states define assault as putting somebody in fear of a harmful or unwanted touching, right? So if I say I'm going to beat your ass, right, and like I pull back, like I cock my arm, I've assaulted you. Like, that's it, right? A battery is a completed assault, right? So a battery is I've made physical contact with you. I've made a unwanted touching, a harmful touching, unwanted touching. I've gone through that action. I've made that contact, right? So that's what we're going to see in homicides, and we're going to see blood in batteries, right? In batteries, I mean, you have everything from bloody noses to missing limbs. I mean, you kind of get the full spectrum, right? So that being said, we're looking at blood. Generally speaking, it's found on or near the victim, right? So that's going to be really kind of one of the main sources, is wherever the victim is, that's where we're going to see blood. If we don't see blood near the victim, it's a pretty good indication that we are actually at a secondary crime scene as opposed to the primary crime scene where the murder occurred, because and again, depends on what ultimately led to the death of the victim, but generally speaking, homicide scenes are very, very bloody. I mean, even think about if you've gotten a bloody nose before. Think about how much blood came out, right? And it's going to stay really kind of in your vicinity, on or near you as a victim. We will also find kind of second most common is points of egress, right? So there's ingress and egress. It's just legal terminology. Ingress is going into a building. Egress is leaving a building or a scene. So we'll find blood probably at a point of egress, of leaving a scene. So if we have a homicide in a house, we're probably going to see blood on the inside of the door, in terms of on the door handle, maybe drops of blood leading to the door, that sort of thing, right? And which is really great because it gives us an idea of how the perpetrator left the scene, which kind of gives us some indication of how they entered the scene. But it kind of helps us follow the clues, right? If blood drops are going to the door and we find blood on the door that belongs to the victim, and the victim's laying there on the floor dead, probably a good chance that that was blood transferred from the perpetrator. The victim's blood transferred from the perpetrator onto the door and the floor. So it kind of gives us really a trail of breadcrumbs to follow, um, and follow them outside and see where the person has gone. We also very commonly find blood in sinks and towels, right? So this is usually when somebody's trying to clean up a crime scene. Um, we're going to see that it's actually very problematic when it comes to blood to try and clean up a crime scene. But yeah, if you find blood, I mean, you're going to check the sink, right? You're going to check, and especially around the drain area, because a lot of people kind of forget, like, blood kind of, the way it works is it starts to harden up and thicken up, and so it does get caught in the drain fairly easily. So we'll find that if we're looking at somebody trying to clean up the crime scene, we're definitely going to look at the sink. We'll find it on towels. This again can be, you got a bloody nose, so you put a towel over your nose to stop the bleeding or to catch the blood. Then obviously we're gonna have blood on the towel. And again, it happens very commonly when we have somebody trying to clean up a crime scene. We'll also find blood on weapons, right? Now, what's interesting about this is the blood on weapons, for the most part, it's probably going to be the victim's blood, right? And then that makes sense, right? Except if we have a stabbing, and we'll talk a little bit more about that when we get into really the homicide specific portion of this course. But if we have a stabbing, there's actually a really good chance that we will find two sets of blood. Right? We will find blood of the victim who got stabbed, and depending on how many stab wounds and what kind of attack it was, a lot of times we will find blood of the perpetrator, right? A large part depends on the handle of the knife, if we'll find blood of the perpetrator or not. Because if we're talking about a knife that doesn't have a grip that protects your hand from slipping, think about it, you're stabbing somebody, so you're getting like this, the instrument you're using is getting lubricated, right? And the more you stab, at some point, if there is not on the hilt, if there's not a hand stop there, or hand rest there, or hand guard there, your hand might slide down the knife because, again, it's lubricated. If that happens, you're going to cut yourself, and your blood is going to get mixed in with the scene. So we definitely find it on weapons. If you shoot somebody, there will be blowback oftentimes, depending on how close you are. Probably you're not going to find perpetrator's blood there, but again, you have a very good chance of finding perpetrator's blood at a stabbing. Again, just depending on the nature of the stabbing and what kind of instrumentality they used. Right? And they're also going to find blood on the clothing of the perpetrator. Right? So it's really difficult to kill somebody and not get at least a little bit of blood on you. Even if you are standing decently far away, you shoot somebody, blood's going to come out. It's going to be in this very kind of almost vapor form. It's kind of the reason why we all wear masks right now, right? Because when we speak, we spit out for like 6 feet. That's why we have that kind of 6 foot rule. We spit out saliva. We spit out, I mean, essentially DNA evidence, but we spit it out. We're not trying to. You don't really notice it unless you have like one of those clear shields on. And so that's why we, again, wear the mask. Well, the idea is kind of the same thing when it comes to committing a crime, committing a homicide, especially if you shoot somebody. That blood is going to blow back and get on you in some form or another. And it may be microscopic if you're far enough away, but it's still kind of that same concept of there'll be some blowback, something coming from the victim back out, even if it's microscopic, that will often get on the clothing of the perpetrator. Now, usually homicides are going to be kind of, they're usually conducted on a very personal level. So you'll generally be closer to the victim if you're the perpetrator, so more chance to get blood on your clothing. If you're shooting them, if you're stabbing them, obviously you're going to get blood on your clothing. So that's, again, a really great source. That being said, when we think about blood, we have to understand how much blood is in the human body, right? So there are about 10 pints of blood in the human body, and that sounds like a lot. Reality is it's actually 1 1/4 gallons, right? 1.25 gallons of blood in the body. So think about a milk jug, and then like a big milk jug, and then half a big, or a fourth of a big milk jug. Taken together, that's about how much blood you have circulating in your body at any given time. It may not seem like a lot, right? Because if you can kind of conceptualize it as a gallon of milk, it doesn't seem like a lot. But if you've ever poured a whole gallon of milk on the floor, you'll see it's a ridiculous amount. So again, it does provide us ample opportunity to collect evidence. So in addition to the DNA that we can get from evidence, and we're going to have a little discussion of DNA in a bit, which is, again, derived usually from the white blood cells. We also engage in the practice of scientific practice of what's called serology, right? And serology is really typing the blood, right? So if you don't know, and I hope you do, everyone has a blood type, right? So we don't all have the same blood type. It's very dependent upon your parents' blood types and things like that. But it's the reason why you can't just give blood to anyone, right? It has to be very specific blood. So if we look at blood typology, the most common that we have is type O blood, and that's about 46% of the population, right? So I'm in that category. I have type O blood. Type O blood is great for donating blood, because people with type O blood, depending on their rhesus antigen, are usually considered universal donors, right? So that means, like, if I wanted to give somebody with, let's say, the second most common type A blood, right, which is about 24% of the population, so a fourth of the population has type A. If I want to give somebody type A blood, with type A blood, the blood transfusion, I needed to give them blood, I would use type A, right? If I use type B, the body would reject it, it would poison the body, and would likely kill the person. Or I could use type O, right? Again, depending on the rhesus antigen, for A. So type A can get type A and type O, right? Unfortunately, type O can only get type O, right? So this is why, because you're a universal donor and only type O people can get type O blood, so you have like less of a shot than other people. This is why if you have type O blood, it's so important that you do donate blood. Even if you're on medications, you can donate blood. Like, everyone knows I'm on a ridiculous amount of medication, and I still donate. It's not a problem. So if you type O, just have to consider that. Just a public service announcement. Then we have type B. Type B is the third most common. That's 9% of the US population. And then we have type AB blood. And this is the most rare, and it's 3% of the US population. Now, the cool thing about if you're type AB, you are golden if you ever need a blood transfusion. Because type A, or type AB, excuse me, are what's called universal recipients. Right? So remember, if I give type A, somebody type A blood, type B blood, it's going to kill them. Right? Type AB, I can give them type A, I can give them type B, and I can give them type O. Right? So, and I can give them type AB. So they can take any blood and it's not going to hurt them. Like, it's going to be a fine transfusion. So O, universal donor; AB, universal recipient. Now, what serology does is it helps us start to kind of narrow down a pool, right, of an inclusive and exclusive pool. So if we type the evidence, and we type blood and we say, okay, we found two types of blood here, right? We found type O and we found type B at this crime scene. And let's say it was a stabbing, right? We found type O and type B. What we'll do is we'll test the victim's blood that we know is the victim's blood from their body. And if they're type O, that tells us, generally speaking, we can exclude anybody who has type A blood, type AB blood, or type O blood, right? Because we're looking for somebody who has type B blood. So that lets us narrow down our suspect pool to about 9% of the US population. Now, that's still a ridiculous amount of people, still a lot, and it can never prove guilt, and DNA can't prove either. And we'll talk a little bit about that, but it helps us narrow it down. So, right, so if we have like 3 suspects, we're going to take their blood, or we're going to take a sample of their blood. And maybe we can narrow it down to 1. Maybe all 3 of them have type B blood. We don't know. So it's a very helpful tool. Then we refine this type A, type B, type AB, and O with the presence of what's called the rhesus antigen, right? So you've probably heard of the descriptions of like, oh, I'm type O positive or I'm type O negative. What we're referring to when we're talking about that is the rhesus antigen, right? So if you have the rhesus antigen in your blood, you are positive. And if you don't have the rhesus antigen in your blood, you are negative, right? So that's why we have blood type O negative, blood type O positive. Now, it doesn't mean anything in terms of your health. It doesn't mean anything in terms of like, if you have it, it's some kind of like virus. It's nothing like that. It's just a part of the chemical makeup of blood. But it helps us narrow things down even further, right? So we say we find at the crime scene, what we think is the offender's blood, which is type O. All right, that's 46% of the population. Then we are going to test it for the rhesus antigen and see if it's type O negative or type O positive. And then from there, that narrows it down from about 50% of the population down to roughly 25%, right? So we're working with smaller numbers that allow us to kind of narrow down suspect pools. So when we talk about blood evidence, let's kind of look about the concept of discovery and collection, right? So most perpetrators attempt to hide blood evidence, right? Most perpetrators, I'll say it again, most perpetrators try to hide blood evidence. Especially they're trying to make the crime scene look staged or whatever, but they're going to try to hide it. That being said, there are still several methods to locate what you consider cleaned up blood, right? So ultraviolet light, which is 415 nanometer wavelength. It's really good for spotting blood, and it's also really good for spotting urine as well. So I have a cat with special needs. She's crippled in the back legs, and so she doesn't always make it to the litter box in time. And so like once a day I'll take a black light or a ultraviolet light, and I'll kind of go through my apartment to see if there's a mess anywhere. And I mean, it glows. It's crazy what it does, like with urine. I haven't tried with blood because I haven't killed anybody in my apartment, and I haven't been killed, but I'm leaving that door open. But yeah, it really does glow. That being said, if we're having a difficult time locating with just the ultraviolet light, we often combine it with chemicals such as luminol or fluorescein. Now, you've probably heard of this, or you probably have seen it on crime shows, especially CSI. CSI, I remember when it first came out, they loved using luminol. They used it everywhere, right? Like, oh, this is cleaned up. Like there's nothing here, and then they spray it, and then it magically like illuminates, which is actually true. Ultraviolet light's not going to pick up blood by itself if the blood is really highly diluted, right? So if you used water to clean up the scene, if you didn't just wipe it down, if you use chemicals, like bleach is a real big problem. But yeah, if it's really highly diluted, ultraviolet light might not pick it up by itself. And so you're going to need a reacting agent like luminol or fluorescein. Now, in CSI, they always use luminol. Here's the problem with luminol. All right, they use luminol, they spray it, and then they use a black light to see if there's blood. Here's the problem. Luminol destroys the properties of blood, right? I'll say that again. It destroys the properties of blood. So luminol basically, it'll show you where the blood is, but you cannot collect it, right? The serology is gone, the DNA is gone, it destroys it. So if you use luminol, you have to use it very cautiously and very sparingly, right? To kind of give you an idea of where some blood might be, and then try and collect it from there. If you just spray out a whole crime scene with luminol and then take a black light to it, you've just destroyed all that evidence, right? So luminol is not great in the terms of like, oh, wow, this is a great amount of blood. Well, if you need to collect it and you spray it with luminol, like you're destroying it. You're not going to collect it. Similarly, fluorescein, kind of the other tool that we use, often reacts with common household cleaning agents and is really of little use depending on the perpetrator's actions, right? So whereas luminol is generally going to find us the blood and should only be used for kind of diction purposes or detection purposes, I should say. Fluorescein does not necessarily destroy the properties of blood, but it will pick up a lot, if not virtually all, household chemicals, right? So God forbid you kill somebody on a wood floor that's been sealed, right? So it doesn't fall through cracks or anything like that. It's been freshly sealed, and you use Pine Sol, like you clean up the blood and then you use Pine Sol to like wipe down the whole living room where this occurred, all of the wood. Fluorescein is going to react with Pine Sol. It's going to show that this whole thing is blood. So it's really of little use, depending on what the perpetrator does. Right? So again, if you're going to commit a crime, you're going to kill somebody, you're going to clean up the mess, use as many chemicals as possible, because it takes a lot to get rid of blood, and you can almost never do it, especially if carpet. So you have carpeting, like you're screwed. It's really hard to get rid of blood entirely. That's why, again, we use things like luminol. When it looks like it's gone, luminol can still show us if it's there. If the more chemicals that you use and the more diluted it becomes, the more likely the luminol is not going to show it, and the more likely that fluorescein is going to show everything and be of absolutely no use. Now, let's assume that we're in the discovery process of dealing with blood, right? And assuming that the blood is not destroyed in our detection process, its presence is confirmed by mixing the substance, so mixing what we think is the blood, with two chemicals. I can never say this. It's fenfentanine. I can never say it. And hydrogen peroxide, right? Mixed together. You take the swab of what you think is blood or an area of blood. You put it in the chemical reaction, or the chemical little vat. And if the swab turns pink, then blood is present, right? So that tells you, okay, stop spraying. We found blood. Now, collection of blood largely depends on the state. Generally, and when I say the state, it means the state of the blood, not necessarily like the Texas versus Colorado or something like that. The state of the blood and where it's at, right? Because blood can be, it can be a number of colors. Blood can actually be green depending on the surface that it's on, right? So if it's dry versus if it's wet. I mean, there's all kinds of states. But generally speaking, it should be collected by a swab, right? And we place it in a plastic tube, right? So we try to keep it free from contamination. And then we refrigerate it. We do not freeze it. If you freeze it, you're destroying the chemical makeups of the blood itself. But if you refrigerate it, the idea is you're keeping it fresh, you're keeping it longer than you would if it was just sitting out, right? So again, this is really one of the only times that we're going to use plastic when we do crime scene investigation. Most of the time, we're going to use paper because we want evidence to breathe. We don't want to destroy evidence. We don't want to destroy DNA or anything like that by sealing off the air and it just naturally kind of decomposing. But blood doesn't have that tendency. So we, again, first thing we're going to do, we're going to swab it. We're going to test it. We are going to place it in a vial, and we are going to refrigerate that sucker really quickly, right? Again, not freezing it. So let's talk about semen, right? And no, I'm not talking about sailors. Semen. So semen consists of sperm, right? Those little head with wiggly tail fellows. Generally speaking, there are 60 to 100 million little wiggly fellows, sperm, per milliliter. All right. So basically what that means is, everyone's different in terms of ejaculation. It depends on when the last time they ejaculated was. It depends on, I mean, there's a lot that goes into play. So if we say that somebody just ejaculates 1 milliliter of sperm or semen, which you will more than that, but let's say that we do, there's 60 to 100 million sperm in there, right? And again, when we're talking about copulation that's designed to result in a child, you only need one sperm really to find the egg, break through the egg, and fertilize it. So I mean, you have millions trying to do it, and the body's going to shut it down. Most of them, so it's going to kill most of it. But you just need a few to get through, and one just to fertilize. So that being said, we have a numerous amount of sperm, and the sperm is suspended in what we call the seminal fluid, right? So not everything that is an ejaculant is semen, or excuse me, sperm. There's a lot of seminal fluid, again, kind of the liquidy form, that contains the microscopic sperm. Now, sperm, again, is like blood in that it can be wet, dry, whatever. In liquid form, so if we find sperm and it's still in its kind of fluid state, liquid form, generally speaking, I mean, most of you know this, it's gray to white color, usually kind of tending more on the white. It can be clear. Again, it means some different things, but generally speaking, we're talking grayish to white. Like it will strongly stand out, and especially what makes it stand out is it has a strong odor. Like it smells like chlorine, right? So if you ever smell sperm, it's going to smell like bleach, right? So let's say that we're doing a sexual assault case, and the same nurse, the sexual assault nurse examiner, let's say it's a man attacked a woman, sexual assault examiner, they're going to look inside the vagina, and if they see like grayish, clear, maybe white liquid, it's probably, I mean, it could be a number of things, but it's a good chance that we're looking at semen. So that's kind of when it's wet. Again, the more it stays in the human body, the more likely it is to be wet. When it gets outside of a body, or when it gets outside of, or even in a body after a very long time, but once outside the body, usually it dries out relatively quickly. right? And so when it dries, it's clear, right? Sometimes, depending on the surface, if it's like a dark surface, it might show up as white, but it's generally going to be clear and it's going to be starch-like. What I mean by starch-like is it takes material and it makes it uh, difficult to deal with in the sense that um, it it makes it the material hard, right? Like that's what starch does. Starch, if you and it's I don't think many people use it anymore, but if you have starch spray in your laundry, it's what makes shirts look very pressed and things like that. It's because it makes the material basically hard, right, or harder than it normally is to maintain a look. Well, semen does the same thing. So if it lands on a material surface, it's going to make that material kind of crunchy, kind of crusty. Like it's going to make it uh, hard or harder than it would normally be. That being said, it's not uncommon though for semen, even though it's usually clear and starch-like, to take on a yellowish hue. So especially, again, there's a lot of things that factor into play. But if you find semen that's a little old, it's kind of the older it gets, the more yellow it's going to be. And really that's just kind of it decomposing. Now, when we're talking about semen, usually we're looking at sexual assault victims. We're looking at and again, I say victim, preferred term is survivor, but in law we say victim. Victim's clothing, usually we're going to find it in underwear, right? So let's say there's a sexual assault, the male ejaculates into the female's vagina. Vagina is going to leak out some of that semen, a large part of it. So we'll find some in the underwear. If the perpetrator used condoms, we're going to find the semen in the condom. And we're also going to probably find semen where the assault took place, right? So we're going to find it on a bed or a carpet, some even tissues, right? People try to clean it up or wipe it off or whatever. So semen is pretty common to find sexual assault cases. If it's just a murder without a sexual assault predicate or rape predicate to it, we're probably not going to find anything. It's really, again, it's very specific. It reminds me of the movie Super Bad, right? And the kids in the liquor store and like he gets like cocked in the face by a robber in the liquor store and falls to the floor. The police come and he's nervous, he's underage, and he's like, well, you're gonna catch him? And the police are like, no, we have no chance of catching him. Like if he had ejaculated and then punched you in the face, then yeah, maybe we have a pretty good shot. But like without it, no. And it's true, right? It's usually sexual motivated crimes where we find semen. If we find semen at a crime scene, like that's going to send red flags immediately, because generally you don't have it, right? Now that being said, we do have to account for masturbatory emissions, right? So not just of perpetrator, but I mean of anyone or of any male, really, right? Let's say that a couple is or somebody, or we'll do it this way: a teenager, yeah, a teenager like is murdered in bed, and we're trying to find out if he's sexually assaulted, and we find semen friggin' everywhere, right? It's most likely it's masturbatory emissions, not a sexual assault, but we still have to treat it as a possibility. We can't rule anything out just because of somebody's age or anything along those lines. Now that being said, spermatozoa, which is one sperm. So sperm is the plural of spermatozoa. Spermatozoa is really rich in DNA, right? That's the whole point of it, is it carries the DNA to the egg. And it may be found via what's called an acid phosphate test. So what that is really it's a mixture, oddly enough, of chlorine, which is what semen smells like, and P30, and then we use an ultraviolet light to detect it, right? Now, most commonly, we're going to use ultraviolet light, as other tests may destroy the proteins and the DNA in the seminal fluid. So if we can just do a black light test and find it, then that's what we're going to do before we have to use chemicals, again, because chemicals will be like luminol in the sense of they will destroy the DNA, they will destroy the proteins in the seminal fluid, so it becomes problematic. To that end, if you've ever taken a black light, like if you ever stayed in a hotel, get yourself a black light, and I promise you, you'll never stay in a hotel ever again. Because yes, they change the sheets and things like that, but like there in most hotels, there is semen friggin' everywhere, like even on the ceiling, which I'm like, how? But okay, it's everywhere. Yeah, turn off all the lights, use a black light, look around, and you will not stay at that hotel. You will not stay at a hotel, period. Every time I stay at a hotel, like I just think like, oh, this is really nasty. Like it could be the nicest hotel, right? I mean, a very ritzy hotel, everything is cleaned down, everything is just spotless and beautiful. You're still gonna find like sperm, like you're still gonna find it everywhere. So that also becomes problematic if we have a murder or rape in a hotel, and we don't find anything necessarily on the victim or in the victim. Trying to locate which semen in a hotel room is the right semen is almost like trying to collect hairs or skin samples. It's really, really, really hard. There's a lot to it. That being said, when we do find it, right, even in a hotel setting, but again, just in general, when we do find it, we're going to take a swab, just like we do with the blood. With the swab, we're going to air dry it, right? So we're not going to put it in anything wet. We're going to let it dry in the air. Once it's dried, right, because it kind of sets everything in place, then we're going to take it and we're going to put it in a clean paper envelope. The reason, again, we want paper is paper has pores in it. There's little holes in paper, like little tiny holes, and it lets the specimen breathe, right? So if it didn't, it would start to decompose very rapidly. If we sealed off the oxygen to get to it, right? So we put it in a paper bag. We do not put it in the plastic bags like you see on CSI. Most of the time, what we're using in a CSI person is going to be using paper bags, right? Not plastic, right? Plastic bags, if you want to put a gun in it, fine. It's got fingerprints. There's no way in hell you're putting a gun, like, no. So you're going to use clean paper envelope. Now, this is at the collection stage. When you get ready to test it, the dried semen should be rehydrated. So kind of brought back up to life, re-wet, and kind of regaining its liquid form, using distilled water. So you don't use any chemicals, you just use distilled water. And then again, you can collect it, right? So if you have the crime scene and it's wet, you're going to swab, air dry it, store it. If it's dried, you're going to wet it, and then you're going to get it on the Q-tip, air dry it, and store it in the clean paper bag. All right, so I mean, it's either way, you're kind of following the same process. So thinking about saliva is really interesting, because our saliva consists nearly entirely of water, right? Again, the thing that is spitting out of your mouth, right, as you speak, that's mostly water, but it also contains mucus, proteins, enzymes, and here's the cool part, skin cells, right? So if we find saliva, then that skin cell analysis changes. So saliva is a great source of DNA. It's a great source. And it's usually found, when we're looking for it, we're going to find it on cigarette butts. Again, that's the most common. Bite marks, cans, bottles, find it on clothing, envelope, stamps, really anything that's going to come in contact with the mouth. We're going to find saliva. And from that, we can extract DNA. There's one trick investigators use, and again, this is just kind of for your own knowledge, but if we're investigating somebody and we want a DNA sample, but we don't want the person to know we took a DNA sample, well, the investigators, when they're doing an interrogation, well, again, we'll talk about this when we talk about interrogations, but they will often ask the person they're talking to, who they want the DNA from, if they want a cigarette, or, oh, can I get you a bottle of water, right, or a cup of water? And the person smokes the cigarette and drinks the water, or whatever they do. Investigator doesn't do anything, does not show anything, does not grab it, because technically that is that person's property, and you need a search warrant and a seizure warrant to get that cigarette butt from the cigarette that you just gave them. However, what happens is most of the time, after you talk to the person for a little while, you let them go, right? Like you say, okay, I appreciate you coming down. We'll be contacted if we need any more information, but you're good to go. Most of the time, people are going to leave the cigarette butts in the room, or they're going to leave the bottle of water. They might throw it in the trash can. They might leave it on the table, whatever it is. At that point, it becomes abandoned property, and then you don't need a warrant, and you can do a DNA test. All right. Prisoners are actually really smart. If you try to interview a prisoner about a potential crime that they committed while they were on the outside, and you do the cigarette trick, or you do the water trick, they will take it with them. Like, they will take the cigarette butts, they'll take the water bottles, they'll take it all with them, because they know that you're trying to get their DNA, and they know that if you take it with you, even if it's like an empty bottle, it's still your property. You still, a cigarette butt, you still need a search warrant. So they'll take it with them. I said prisoners, they know the game, but people who don't know the game, and they take the cigarette, right? If you ever get interrogated, don't take the cigarette, don't take the water, don't eat the food, don't anything, because that's their way of getting your DNA. Now, that being said, when we think about saliva is really invisible to the naked eye, right? Again, unless you wear one of those face shields, which I do when I do live lectures, and at the end you see just this nastiness, like of just like, yeah, on the interior of the mask that you have to clean off. But generally speaking, beyond that, it's invisible, right? That being said, saliva can be detected with chemical tests that identify the alamase, which is the melase, which is the enzyme that's present in high concentrations of saliva, as well as other types of tissue and bodily fluid, right? So again, when we're looking for saliva, we are going to want to use an alternative light source. Again, preferably UV light, to see if we can detect it, because depending on the chemical test, and then we have two problems, right? One, if we don't have a high concentration of saliva, if we just have a low concentration, chemical test might not pick it up. And depending on what chemicals we use, we can destroy the DNA in it, right? So it doesn't really help. So something like and some stuff is obvious, right? So if we have a bite mark on somebody, we are not going to use chemicals. Maybe we'll use black light, but probably not. Like, we're saying there's a really good chance that there's saliva here. So we are going to swab all around the saliva, or all around the bite mark, just to see if there's something there, right? And there probably will be, because again, yeah, that transfer from the mouth. So the collection of saliva is the same as the collection of semen, right? So you take it, if it's wet, put it on the end of the Q-tip, let it dry, put it in an envelope. If it's dry, rehydrate it, put it on the Q-tip, let it dry, put it on the envelope. And the same is actually true for urine. Urine is an interesting case because it has its own properties. So urine is fairly rare at crime scenes. Like, it's not something that really happens a lot. But urine does contain, it is sterile, right? So it does, though, contain a lot of information. Um, so when your body gets rid of like blood cells, because your body's constantly making blood cells, and the blood cells are doing their job and then they're getting damaged or they're getting old or whatever, your body flushes them. So like every time you urinate, you're flushing out red blood cells, you're flushing out um, DNA galore, but it's, again, it's sterile. It's not like it can infect you or anything along those lines. So if we find urine, we do the same sort of collection. And again, it's really rare that we'll ever even look for urine. Even during sexual assaults, we're probably not looking for urine. If we find it, then yeah, then that's great. We can try and use it and try to see what we can get from it. But it's really rare. Unless you're like, oh, the perpetrator went to the bathroom before or used the toilet at some point during the attack. Well, in that case, you might try to find urine, because no matter how urine, let's say you're male and you urinate, most males urinate standing up, which causes blowback, right? So hits the toilet and bounce like microscopic amounts bounce back and like get on your clothes and get on the floor and get on everything, right? Same true for feces. Like if you ever have your want to be grossed out, have your toothbrush tested for feces. If you keep your toothbrush in your bathroom, it will be covered in feces. So just know that every time you brush your teeth, you're brushing your teeth with your own feces. So there will be blowback, right? And then, again, most women urinate sitting down. There will still probably be blowback, not as much, and in different locations than there would be for a male. Then we might look for urine, but again, it's fairly rare. So then we have hair. So hair is most likely to be found in cases of a struggle, right? So we are talking if the victim put up a fight in a homicide or a battery, then we're most likely going to find hair. Right? Now, it can often be found on the victim or the perpetrator's clothing. We're going to find hair in vehicles, on personal items, and on cleanup sites. So you clean up the blood, you still shed hair. You shed hair just like you shed skin cells. Now, that being said, hair is very problematic, right? Because the only time that we are going to get hair, DNA from a hair, is if the hair has been ripped out of the person, of the flesh, right? So hair that just kind of falls out, just sheds, we're not getting DNA from. It's a collection of dead cells, we're not getting DNA from it. But if you pull a piece of hair out of your head, or you pull a piece of hair out of your arm, or you pull a piece of hair out of somebody else's head, what will come with it, and we'll talk about this in a second, is one of the parts of the hair follicles, the root. The root is where the nuclear material is stored. So that's where we're going to find the DNA. When you shed hair, the root doesn't leave, right? The nuclear material stays in that hair pore. But again, if there's a struggle and you pull it out, then you're probably going to take the root with it, and then that will give us DNA evidence. Now, that being said, hair has been very problematic in the past because there's significant variations, right? Even from a single individual, right? So I just love the typo there. Think about this, right? Think about the hair that's on your head versus hair on your face. And yes, ladies, you usually have thin hair on your face too, versus your pubic hair, right? Like they are all different for the most part, but they are on one person, right? So hair varies on a person. So hair, by definition, varies from person to person, right? I mean, like crazy. There's a big variation, and it's been problematic in the past. It has had some racial components to it. So again, different races tend to have different their hair, all of our hair consists of 4 parts, right? The cuticle, the cortex, the medulla, and the root. So all hair consists of that, but how it looks, how it forms, how it shapes, the color, everything like that really depends upon lineage, depends upon race, and things along those lines, right? So, and you can't say all, but it's significant. There was a problem when we used to have, and we still do to some extent, sexual assaults, all right, or assaults in general, or robberies, or whatever. You might find a hair and be like, oh, this is a pubic hair. And no, because it's short and it has some curl to it. It's dark in nature, so it has to be pubic hair. No, that could theoretically belong to an African American, right? Again, their head hair tends to have curl to it, tends to be short, just depending on if they're male or not, and tends to be dark in nature, right? So we can't just look at a piece of hair and say this came from this race or this came from this place on this person. We can't do that, right? So the hair, the thing that we need, the thing that's the most important part of the hair is going to be the root and the DNA and material in it. There have been many wrongful convictions based upon hair evidence because we've said, oh, we found a short curly hair, so it has to be a black male. No, doesn't. You can't distinguish between hair of a black male and pubic hair. You can't distinguish between like long hair of a female and, I mean, depending on how long the hair is, and hair on the arms. It's just, again, you just can't. And in the past, we've tried, and we have convicted people wrongfully because we're like, this hair looks like their race. And you're like, that's not, no, you can't do that. Yes, races do have typologies, but again, it's hair's tricky. It's not what you think it would be. So that being said, I'll just quickly talk about the 4 parts. So we have the cuticle, the cortex, the medulla, and the root. The cuticle is the outer core of the hair, right? It consists of overlapping cells. So if you ever take like a look at a hair under a microscope, you're gonna see like it almost looks like armor, right? That's the cuticle, that's the outside. Then you're gonna have the cortex. So the cortex is underneath the cuticle. It's what's gonna have the pigmentation, right? And pigmentation determines your hair color. So it's gonna be black, it's gonna be white, it's gonna be brown, it's gonna be whatever, right? The cortex contains the pigmentation. And then we have the medulla, which is the inner layers of the cell. This is often destroyed by hair dye. So ladies and gentlemen, I mean, this is true for you too. Whenever you dye your hair, you are doing massive amounts of damage to it. You are actually destroying the hair itself, the chemical makeup of the hair, the physical makeup of the hair, you're destroying. And I know this because I went to, okay, to be fair, I went to high school like when boy bands were popular, right? Like NSYNC and things like that. And did I have frosted tips? Yes, I did. Right. So yeah, you can picture me with like frosted tips. Like I had frosted tips. And I was taking a class and we looked at hair underneath the microscope to see like the different parts. And if you look at hair underneath the microscope, you can usually see the cuticle, the cortex, the medulla, and depending if it's been ripped out or not, the root. Looking under the microscope, I saw the cortex, I saw the cuticle, I saw the cortex again, it had been altered, but there was no medulla. Like it was destroyed, it was gone. Like it was just cuticle, cortex, and really chemicals. So again, that's another problem with hair, is it can change colors, and it's not necessarily, if you dye it, it can hurt it. And it can change a whole lot of things, right? Again, even thinking about like the pigmentation, right? Let's say that you're a blonde, chances are that your pubic hair is going to be dark in nature. Could be blonde, right? Let's say that you have red hair. You probably, well, actually, redheads have a little bit different. redheads don't have souls. We all know that. But they also, their pubic hair generally tends to be red as well, although it can be dark. So we can't ever really make like sweeping assumptions. And yes, the redhead thing was a joke. Like, they have souls. Just in case you have red hair. So that being said, when we do hair testing, we look to the root, right? Because the root has the DNA evidence. That's where the nuclear material is located. Now, generally speaking, again, this is kind of a little bit repetitive, but you shed between 100 to 125 hair follicles, so pieces of hair, per day. It's unlike the skin cells where you brush your hand and it's invisible, they're microscopic, but you have just tons falling off, right? So you only have about 100, 125, which is still quite a bit, right? And for the most part, unless you're balding, you probably don't notice it, right? You might find like a piece of hair in your clothing and like pick it off real quick, but you're not thinking like, oh, that came from me. Like, it's just a piece of hair. The hairs that fall out, none of them contain a root, right? The root can only be removed by extreme force. So hair pulling during a struggle, right, is where we're going to have it. Or if you want to look at what a root looks like, take a piece of your hair, from an arm, from a pubic region, from your head. It's going to hurt. Yank it out. You're probably going to get a couple. You can even get like hair in your ears, and yank those out. And what you're going to see is at the very end, the very tip, you're going to see this like little white ball, right? That little white ball is the root. That's where we can get DNA. The only time that white ball comes out is if that hair has been pulled, right? So this is why, if there's evidence of a struggle, then we're going to start really looking for hair, all right? There's not evidence of a struggle, then hair might not necessarily, it's not going to be really that useful. Now, that being said, there becomes questions of, well, do you still collect hair if you find it? And the answer is yes, because if we do a chemical examination of hair that doesn't have the root, right, so we don't have the DNA, but we do a chemical examination, it can reveal the presence of drugs or other ingested items, and a neutron activation analysis can measure trace elements in the sample, right? So this is true of if you want to be a teacher in the state of New York. When I taught at the University of Alabama, I didn't even have to take a drug test. Like, to teach up here, the state of New York requires that you take a drug test, but more than just like a urine drug test, you have to take a hair drug test, right? So your hair has to be a certain length. They cut your hair off, and then they do chemical analysis on it and see like what's in your hair, right? So that's why when you think, like, if people say, oh, you smoke marijuana, if you're a heavy smoker of marijuana, it's going to stay in your system probably about 8 days, right? If you're a light smoker, it's probably going to stay in your system a day, if that. So urine tests aren't going to generally pick up marijuana, again, unless you smoked within the last 8 days. But hair tests will. It doesn't leave the hair, right? Now, it's not to say it doesn't leave the hair forever. It doesn't leave the hair until it's grown out and that part has been cut. It's almost like a tree. If trees have those rings, and like the rings kind of tell you how old that tree is. The same thing is true for hair, right? As the hair grows, little rings that have like marijuana residue and stuff like that, they get further and further up the hair, and then you cut it, they're gone, right? Some stay longer, some are more present than others. So generally speaking, harder drugs are going to be a lot more present and a lot more likely to be detected. But yeah, if you have to take a hair test and you've done drugs like within the last 2 or 3 months, because that's usually about how far back we can find out if somebody took drugs. It's about 3 months. It's probably going to show up. So yeah, just know that. And don't do drugs, kids. That being said, how do we collect hair? Well, we collect hair in a fairly kind of obvious manner. We use tweezers to take the hair. We take this single hair, and we have to take a single hair, and we place it in an envelope, right? A paper envelope where it can breathe. But we have to seal the corners, and we seal the corners so the hair doesn't fall out of the envelope. We do this for every single hair that we find. We cannot mix hairs together, because we start mixing hairs together, we might get, we'll contaminate the sample, because we'll be getting hairs from everybody, right? That's been in a room. We're going to get hair from them. So you have to do every single piece of hair individually in its own envelope. Now, again, investigators have to be careful, as courts are becoming a lot more skeptical of hair analysis, following a bad science, bad science and wrongful convictions. I'll say this: when my hair test, when I did my hair test to come teach up here in New York, I was ready. Like I was like, I'm ready for the phone call, because part of the medications I take a ridiculous amount of Adderall prescribed, and I take Clonopin, ridiculous amount of that as well. For anxiety, for ADHD, for depression. I mean, like, it's just, I take a lot of medication. And my hair, I keep it relatively short, so like it was barely long enough for them to do it. But they did it, they cut it, they went to the laboratory, and I never heard anything back. Which means they did not pick up the fact that I had Adderall in my system. And Adderall is basically like legalized meth. It's amphetamines. That didn't get picked up, nor did my Klonopin. I never got a call about it or a question about it. But the other day, I'm taking up a part-time job just to help out at the hospital, right? Because it's such a hard time for them right now. I want to help out with them. I do a urine test, and it picked up my amphetamines, my Adderall, but it didn't pick up my Klonopin, right? So again, and the reason I kind of use that as an example is really chemical analysis is a crapshoot, right? Like it's not good for court. It can tell you some things, but I mean, oh, this guy used pot in the last month. Yeah, you probably just described like 90% of America. Like it's not that helpful, and you're probably not getting everything that you need to get from it. So hair is really, really problematic. So let's kind of break down, in all these things we're looking at and trying to get DNA. So let's kind of break down what DNA is. So there's two types of DNA, right? You have mitochondrial DNA. Now that is found in each cell of the body, and it's much more limited than traditional nuclear, what we think of DNA as nuclear DNA, because mitochondrial DNA is inherited from the mother only, right? So you get your mother's DNA in your cells. The problem with this is it becomes really difficult to differentiate DNA, mitochondrial DNA, if like you have suspects that are in the same family, right? So a brother and a sister will have the same mitochondrial DNA. They'll have the same DNA from their mother. They'll have the exact same mitochondrial DNA. And if we're like, well, there's two suspects that could have killed this person, the son and the daughter, we have DNA, but it's mitochondrial. Well, it's not going to tell you a thing, right? Because they're both going to have it. On the other hand, what we want is the gold standard, and the gold standard is nuclear DNA, right? So nuclear DNA, that's the traditional, when we think of it as, it's only found in the nucleus of a cell, right? And it consists of billions of base pairs. We'll talk about what that means in a second. And when it's structured like the double helix, right? So everybody kind of knows that picture of DNA where it's kind of like that little like corkscrew looking thing. That's the double helix, right? And so DNA really has three parts. It has, and I can never say these, adenine, cytosine, guanine, and thymine. So they're abbreviated ACGT, right? These are the chemicals that make up your DNA. These are the proteins that make up your DNA. So they do have natural rules that they follow. So again, base pairs means that the A, C, G, and T, they pair up, right? So you'll have one side of the helix, one protein, on the other side, another protein. And most DNA is the same for everyone. There's parts of it that are different that distinguish us. But yeah, it's going to be mostly the same for everyone. So if we think about kind of molecular biology, a nucleated cell, so one that we can get DNA from, consists of three main parts, right? It consists of the nucleus, the cytoplasm, and the cell membrane. So the nucleus houses really tightly wound DNA structures called chromosomes, right? Now, cells generally have 46 chromosomes, and they're divided into 23 pairs, which are base pairs, right? Which are those letters, ACGT. Now, pairs 1 through 22 are called autosomes, and they govern the cell structure and the function of the cell, right? Pair 23 determines the gender of the individual, right? So again, we're looking at DNA, and we're kind of, it's basically a code of people, right? Of like, okay, this is what the cell does. This is the da-da-da-da. This is how this person's sex or their gender is going to be, sex is going to be, so on and so forth, right? So this is when we think about like the XY chromosomes, right? So contrary to popular belief, you cannot control whether you have a baby boy or a baby girl. Like some people are like, well, you have to keep you have to do it in the hot tub and you'll get a boy or something like that. No, and ladies, unfortunately, it is not up to you what gender gets passed on, what your kid's gender will be or kid's sex will be. It's actually up to the male, because women have double Y chromosomes. So they have two chromosomes, they have YY. Males have XY, right? And each parent donates basically to the child one set of their chromosomes, right? Now, the female, there's only Y's to choose from. So it's either going to be a Y or a Y. But from the male, it can be a Y, which will result in a female child, or it can be an X, which will result in a male child, right? And there's no way of really controlling that. But generally speaking, if you see families where like they all have like, there's like 9 daughters or whatever, it's because that man is passing on that Y chromosome more so than the X chromosome, right? So that's what pair 23 does. So nucleus, that's kind of like where the DNA is, and that's where our codes come from. The cytoplasm basically contains the other parts of the cell, such as like the organelles. This is we'll get the mitochondria organelles, and they have their own small DNA component. But again, we're talking about mitochondrial DNA, so it's just going to tell us a family lineage. And then really the real big part of the cell is the cell membrane, which is the exterior barrier of the cell, which is actually in and of itself an active organelle. So again, as I said before, DNA presents as a double helix. So it resembles like a twisted ladder or a corkscrew, right? I mean, you all have seen it. You all know what I'm talking about. Now, the sides are comprised of alternating sugars and phosphate groups, right? So again, if we think about a ladder, right, the ladder's got like two wooden poles, right, on each side, and it's got like slats in the middle, right? So the 2 wooden poles on each side, they're sugars and their phosphate groups. The rungs of the ladder, so the slats of the ladder, this is the important part, they're the ones that are comprised of the base pairs. Now, base pairs do not appear at random in DNA. Instead, base pairs always occur as follows. A will always, always, always be paired with T. C will always, always, always be paired with G, right? We're not going to see a deviation. Like that is how DNA works. And the thing is in determining, like when we do DNA testing and we're looking at base pairs, we're looking at like the order, right? So it could be like an ACCCACCCCCCA versus a CCCAAACCAAA paired with their respective partner T or G. Now that being said, these pairs, right? These proteins, they are held together by a weak hydrogen bond. Now the hydrogen bond can be easily disassociated and divided into two separate strands, right? Now this is a critical factor for DNA analysis because we only find like one set where, well, where's the other, right? That something caused a break in the DNA ladder, basically. So it's a very critical factor for DNA analysis. So, as I said before, 99.9% of DNA base pairs of each human are the same, right? However, again, the order of the AT and the CG represents distinct, repeats, the way it repeats are distinct across individuals, right? So thus typing requires particular locations, which we call loci or loci, to identify a pattern. Now the FBI uses a 13-loci DNA profile as its standard practice, in which we have 13 locations that we examine because these are where that 0.1% of the DNA is going to be different. Now, that being said, the 13 kind of locations, that's kind of the gold standard right now. Chances are, now the contrary to popular belief, based upon our 13 locations, right, and the profile that we generate and the pattern that we get from those locations, there is a 1 in 1 billion chance that somebody shares the exact same DNA. So think about it this way. Hypothetically, and this is more kind of theoretical than like actual, but theoretically, at any given time on this planet, 7 other people share your DNA. They have the exact DNA profile you do. Now, as a defense attorney, I always like try to point that out, but then you're like, dude, you limited it down to 7. Well, we don't even know who the other 7 are. But yeah, I mean, try to argue what you can. But yeah, at least in theory, 7 people on Earth share your same DNA. So there's somebody that looks like you, has the same issues you do. I mean, like everything that is genetic to you, they have, right? So yeah, it's a little different than what you're taught. Now, as a result, because we can't say 100% certain that a person committed a crime, DNA cannot tell us that, right? Because there's 7 people on Earth that are going to share the same DNA, at least statistically and theoretically. So DNA can only be classified like other non-identifying pieces of evidence. So DNA can be inclusive, exclusive, or inconclusive regarding an individual's membership to a DNA, right? So what they'll say is, okay, tell us about the DNA on this, and then the tech will talk about the DNA. And, well, did you reach a conclusion? I did. What was your conclusion? Well, based upon the base pairs and the ordering, the defendant it's consistent with the defendant, or it's inclusive of the defendant's DNA, right? Which means he probably did it. It's exclusive of the defendant's DNA, means you probably didn't do it. Or it's inconclusive, right? The hydrogen bonds were broken. We can't tell. We're not sure whether they belong to this group of 7 people on Earth that have the same DNA, right? So it can never point to an individual, but instead it can point to a very tiny, 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 tiny group of people. Again, largely theoretical, but it can never point to a person. So DNA is not identifying, right? It is not personally identifying. It can be grouped, but it does not and can never identify a single individual. And something definitely to remember. So in terms of DNA collection, The human body has 37 trillion cells. Each contain a nucleus except red blood cells, right? So again, that's part of what comes out when you urinate. It's all red blood cells, so like that doesn't help. But because there's 37 trillion cells, it provides ample opportunities for collection, right? So we're talking most common types of cells used for DNA profiling are white blood cells, right? So we find blood, we're looking for the white cells, epithelial cells, so basically the cells that line the inside and outside of our bodies, and sperm cells, right? Those are our things that we're really looking at when we're looking at DNA. Now, there are three forms of DNA collection. So there's swabbing, cutting, and recovery. Now, swabbing is where you take the swab, and we kind of discussed how you take a swab and you wet the sample. If it's not wet, you just take it and dry it, put it in the envelope. If it's dry, you wet it, then you take it, you dry it, put it in the envelope, etc. Cutting is removing something that has DNA on it, right? Or that you think has DNA on it. So let's say that there's blood on the drapes. You're not going to take the full drapes into a crime lab. You're probably just going to cut off the part that has the blood, right? And then there's, again, recovery of an entire item. So maybe you do take the entire drapes in, right? Maybe it's important for some reason in that case. Now, our goal is really eliminating cross-contamination, right? So cleaning solutions and cutting swabs is critical because of the tools used during later analysis, right? So again, we try to stay away from chemicals as much as possible because they will get destroyed, especially during the analysis portion. We could generate more DNA cells, but the original cells will get destroyed. So that being said, kind of on that note, when DNA is lacking, right? When we only have a little bit and we need a lot to be able to study it and to be to group it, we can replicate DNA using a polymerase chain reaction, and we can create 100 billion copies with a PCR. So PCR has 3 steps. The first step is you take the stranded DNA and you have to separate it into single strands. And you do that by heating it to 94 degrees for 45 seconds or longer to separate it, right? Because basically it makes that hydrogen bond dissolve or vaporize. Then you take small segments of the single-stranded DNA, so basically 20 bases in length. These are called primers, and they're constructed to bind to specific DNA regions by cooling the DNA, right? So you heat it, then you cool it, then the primer is extended at an optimal temperature using what's called tag polymers for the duplication of DNA, right? And what you have to do is you have to do this process approximately 30 times in order to get sufficient DNA to get a template, a visualization to make a profile from. Now, I don't expect you to understand all the science behind this. I don't expect you to understand like the tag polymers and what the primers do, but just know that you have to separate it, use heat, you use cooling, and it replicates. And when it replicates, we have to do about 30 cycles, and then that will get us about 100 billion copies. Then we can generate a DNA profile. So DNA storage and testing. So DNA should never be stored in a plastic bag because it will retain its moisture, and the moisture will break down DNA, right? Water is a universal solvent, which means water, given enough time, can dissolve anything, right? It's a universal solvent. So if you let something retain its moisture in a bag, it's going to destroy the DNA. You put it in a paper bag, it's going to breathe, right? And that moisture is going to get out of the bag, right? It might be absorbed by the bag, or it goes through the pores of the bag. It's going to be able to breathe. Now, that being said, while comparisons are difficult if we have an unknown suspect, we do have something called CODIS, right? Now, CODIS is the government's DNA database, right? And presently it has roughly, and this has actually increased quite a bit, but roughly 12.5 million offender DNA profiles, right? And an additional 2.5 million arrestee profiles. So basically what happens is when you get arrested in some jurisdictions, they take your DNA, they take a mouth swab, and they put it in a CODIS, right? Other ones, it's when you've been convicted and we go to prison, they take the mouth swab and get to the CODIS. And the idea is then they can run it and see if you've done anything else, right? Like if you match with anything else, but it's also there for the future. So if you do something in the future and they find DNA, they can run it against CODIS and see if there's the matching in the base pairs. Now, this is used by more than 50 countries, and virtually every jurisdiction in the United States, even the smallest one, will do DNA collection. Now, it is rapidly becoming a crime-fighting tool, but it does push the boundaries of the Constitution. So I highly encourage you to take a look at this link. It's a link to a video, really kind of questioning whether it's ethical or legal or not to take an arrestees, not a confirmed offender, but somebody that we've just arrested. It can be for drunk in public. We're still going to take their DNA, whether it's constitutional. So that being said, I'll just finish up with this. DNA evidence is usually destroyed 2 to 3 years after a trial, or approximately 5 years after a defense when we have no suspect. Now, both victims and offenders are pushing for DNA preservation and access laws, right? Because basically offenders want the DNA to exonerate them if it's present and it doesn't match theirs. 50 states have post-conviction DNA testing, but it's really difficult to get. So in New York, you can get this for offenders after a trial. You can apply for post-conviction DNA testing. At any time after the entry of the judgment, defendants convicted after the plea of certain felonies must meet certain additional criteria to be eligible for testing. Right, so we're talking about 5 years, pretrial demands, things along those lines. So there's really specific legal requirements for offenders to get DNA. Now, so for survivors, the DNA is really important preservation-wise, especially when it comes to rape kits. Right? Because when there's a destruction of rape kits, as there was in New York City when the hurricane hit, it ruins the case. Like it's over. So it can have dire consequences. And this is a link to another video. It's Samantha Bee, but she's a comedian, but takes a very serious take upon rape kits and their storage and kind of how offensive it is that we don't treat these major pieces of evidence with the care and the respect that we should. So that being said, next class, we're going to look at non-biological evidence. So whether this is going to be like our trace evidence, our digital evidence, things like that. Then we will move on after that to interrogations, confessions, and then we will move into the bloody, gruesome, gory, practical homicide investigation book. So as always, if you have any questions, please feel free to email me, text me, call me, whatever you need to do. I'll be more than happy to talk to you about it. That being said, I hope you have a great day. And if you need anything, again, always let me know.